words which I'm not sure whether we can display. So let me just write up the problem. Just to remind us, and then I'm not sure if I can make the PDF show us the bit we need to see. There must be a way to make it go gradually. Sorry, I'm not very good with PC computers. So we have to consider what approach to use, and I'm suggesting this one to practice using the phenotypic fitness approach. And so first of all, I just go through quickly a little bit about that approach. And this is all taken just from the textbook, so it's all in there too. This is just a shortened version. But the idea is that we have phenotypes, and we could call the phenotype of individuals I for different phenotypes, different values of I. And we want to determine the fraction of the gametes that are contributed by adults of the different phenotypes in the parent generation contributed to the next generation. And that's really, that sentence is repeating that same thing. And we want to take account of the contributions, obviously, through male gametes and female gametes, and we can call those WIF and WIM. And the different phenotypes in the population could affect those fitnesses already, because individuals are mating with other individuals. And so the fitnesses are not, it's not a simple thing like saying, you know, survival or assigning a selection coefficient. The fitnesses emerge from what we assume about the rest of the population, but another important thing that determines them is the mode of inheritance. So I already gave you the example of cytoplasmic transmission, maternal transmission of some genetic factor that causes the phenotype, and then the fitness in question just depends on the female part of the contribution to the next generation. Whereas with autosomal inheritance, I should have probably written them the opposite way around, you have the offspring, each offspring gets a contribution from a male parent and a female parent, and in the whole population, every individual has a male and female parent, so that the fitness contributions altogether will be the sum of both of those for the population as a whole. So in order to use this phenotypic fitness model or approach to looking at this question, we assume that there are, even though we have a genetic variant, so we can have in a diploid 3 different genotypes, A1, A1, and the heterozygote, and A2, A2 for the other allele, the number of phenotypes in the system is assumed to be only 2, and then we can compare the fitnesses of those phenotypes. Now let's see if I can make this go down. Yeah, so that's the assumption, and just a note that I didn't mention in the lectures, but it can be shown that in general when this approach will work and when you shouldn't use this approach. And here are some references about that if anybody is interested in that. And in the present case, we need to assume that one or the other of the alleles is dominant, and this was first done in a paper that we published, Brian and I, a long time ago, in which Brian also added the proof that in this case it gives exactly the same result as you would get if you worked out the full population genetic details of calculating how many offspring of each genotype are produced and calculating the allele frequency change. And in either case with this simplified approach, you'll get the same result as with the full genotype frequencies being carefully written out and correctly, the algebra done correctly, that the new phenotype, if we want to call it a new phenotype, phenotype 2 will increase when its fitness is greater than the other ones, and an equilibrium will occur when they both have the same fitnesses. So what we want to assume for this particular case let's assume that A2 is dominant. So this is going to be, like I said in the lecture, a dominant female sterility mutation, so that A1 individuals are the only phenotype that would be co-sexual, and the heterozygotes or the homozygotes for A2 would be males. And so we're asking female sterility mutation, so that's a male mutant. Right? And obviously, in order to do this, we have to make some assumptions about the phenotypes. And so we're going to assume selfing rate, S. I think I can probably leave these assumptions up, so I don't need to write them. So they have a selfing rate, which we could write for S, so we're assuming that mixed mating model that I mentioned. We're assuming that progeny, if they're produced by selfing, suffer inbreeding depression, so their fitness is reduced by an amount delta relative to the fitness of outcross progeny, so the fitness of outcross progeny would be 1, and the fitness, which here we mean survival or something like that, of progeny from selfing, which we often call selfed progeny, means the progeny produced by selfing, would be lower. Like that. Are those assumptions clear? What I mean by the survival? Somebody looks if they've got a question over there. Are you all right? Or you've got a question? Okay. And then, of course, we have to assume something about the reallocation of resources, or at least about the male gamete output, the male fertility. So we could assume that males produce 1 plus something male gametes, whereas the co-sexes just produce 1. So we're looking at an increase relative to the co-sexes. Everything works the same, you just deal with the relative values. So now the problem is, as I said, to calculate the invasion conditions for a female sterility mutation. What is required in order to have the males have a higher reproductive fitness, or higher fitness than the co-sexes? That's what I'm asking you to work out. So we might go back higher up. We're going to assume this autosomal inheritance, and we need to calculate these 2 pieces to get the fitness. And we need to calculate it for the 2 phenotypes, right? In order to compare and ask whether phenotype 2 has a higher fitness than phenotype 1, the co-sexes that were there beforehand, let's say. Matter. Is that clear? If anybody doesn't understand what they're doing? This is very simple to do. The answer. I need to see how you're getting on. I'll leave you a few moments. Phenotype 1, the co-sex female. Contribution to the next generation. It's a good place to start. What does the co-sex contribute to outcrossing? Yes, it can contribute as a female. It can have offspring through outcrossing. What's the probability of outcrossing? Yes? Mm-hmm. The co-sex has a probability of producing offspring. Did I write the self-fertilization rate down? What? Yeah, 1 minus S, that's right. So it has a probability of 1 minus S of producing offspring by outcrossing as a female. We're just looking at the female contribution to the next generation. So every outcrossed progeny it produces will have 1 gamete from that parent, and the probability that a progeny is produced by outcrossing is 1 minus S. So S proportion of its female gametes are self-fertilized and 1 minus S are outcrossed. Okay? And what about self-fertilization? So it can also produce offspring as a female through self-fertilization. So we have probability. Yes? And how many gametes go into each progeny produced by selfing? 2. So we have 2S. And is that all? Are we going to count 2S? Anything else? Yes, we need to downweight those. We need to discount them because some of them have suffered some inbreeding depression. So we have that. That's right. What about W2 of S? The new phenotype, the other phenotype. What's its contribution through? So this is phenotype 2. Zero. Yep, everybody happy with that one. That's a nice, easy one. This doesn't go up. W1 through male reproduction, how can it contribute gametes to the male, to the next generation through male reproduction? It has to contribute them by fertilizing the female gametes of the phenotype that does have female gametes, right? Yep. So what proportion of their gametes are available? 1 minus S, yeah. It's not 1 minus delta, it's just 1 for, I think that was just a slip. The fitness of the self ones is 1 minus delta. The fitness of the outcross progeny, which is what these ones would be, that's 1. I don't have to write really, but just to make it clear. Right? It can contribute gametes only as a male, only by fertilizing ovules, and this other phenotype doesn't have any ovules. But we have to consider that some of the gametes, some of the male gametes will come from the other phenotypes. This is now not independent of the rest of the population. So we're going to have out of these 1 minus S, some of them will be fertilized by pollen from this kind, and some of them will be fertilized by pollen from phenotype 2, which is male. So the frequencies would come in. So we have to have a frequency we could call it X, males. So we're going to have that plus, of males, sorry. We're going to have 1 minus X, let's start with. We have to divide by total male gametes that go in. These have output 1 of male gametes, and then the other ones have 1 plus K male gametes, like that. I think that's right. It's difficult writing on the blackboard. If I do something idiotic, just tell me. And now I haven't left room to put the male fitness of phenotype 2. See if I can make a little space for them there. They're competing for these same female gametes that are not self-fertilized, right? We have that. Over the same thing at the bottom, right? So now you'd have to work out the total fitnesses of each of those 2 types. That's very simple. And what are we going to do to consider the invasion of males? This is the regular, it's not a trick, it's a regular procedure that we do in these sort of population genetic or evolutionary questions. We say, let this one be at low frequency, so we're only interested in when it's very, very rare. Okay? So you could work out the fitnesses of the 2 phenotypes when males are rare. See what condition you get out of it. I think I want to see a condition that says what value of 1 plus K do we need to have? Yeah, all this hard work is making everybody hot, so we have the air conditioner on. So I'll make space for the answer while you work it out. Did anybody find an answer yet? Oh, I'm being stupid. I did. No, that's right, it's 1. But I did make a mistake. I went too fast here because in the female, I gave it a contribution of 2 through selfing. One of those is through female, but the other one, of course, comes from the male contribution of the individuals that can self-fertilize. I should have put 1 here and 1 in the bit of fitness contributed through male reproduction. I was jumping a step I shouldn't have jumped, but it doesn't matter because I didn't put it here. I put 2 of it there instead of 1 in each place. I hope it's still right. Okay, so I'll tell you the answer, at least I can see where I wrote it down. I want to write it down right. We have to have this greater than 1 minus S times the inbreeding depression over 1 minus S. Did anybody get that? Yeah, I could see a couple of people nodding. Let's hope. Otherwise, you can play with it. What you have to realize is that we're looking at this males being rare, so we don't have to worry about the frequencies. We are assuming that the co-sex is almost the whole population and very close to that situation where the males are coming into a population where co-sexes don't exist. Everything gets very, very simple immediately, and you come up with this result. So now let's just look at a couple of questions that I put down going to here. We had a different formula for the increased fertility, female fertility that females have to invade. If you compare this one with that one, obviously we could put this in terms of K if you like. That's very simple, I won't do it. But there's a different result for male, invasion by males and invasion by females, right? So what would be the result for females if there was no self-fertilization, if we are completely outcrossing? Taking this one. What would you have if you had a completely outcrossing population for the invasion of females? That's very, very simple from here. can hear people saying something. Say it loud. What happens if S is 0, we have complete outcrossing? Yeah. It's not a trick question. It's very, very simple. Yeah, what do you get then for little k? Yeah, speak loudly, please. Sorry? No, there's no 2 in it. K has to be greater than 1 if S is 0. Yeah? This is little k here. For females. This is the condition for females. We should have called them something different, like A and B, but we call them big K and little k. Big K was the one we just derived for males, but I'm saying it's a different condition for females, and that's the one. Right? So you have to have little k greater than 1, which k is the increase in female fertility compared with the co-sexual, and so k having to be bigger than 1 means you have to double. Maybe that was what you were saying, but it's not 2, it has to be double the co-sexual. Now how about this one, when there's no selfing, what do you get? It's the same. Yeah, that's right. So in that case, it's the same. That's right. Now, supposing now there is some selfing, what does it do to this condition? Does it make it easier to have some selfing, or does it make it harder to have some selfing? Does it make the value of k that satisfies this inequality, does it make it bigger or smaller? Makes k smaller? Makes the bottom here, this 1 minus s, when s gets bigger, 1 minus s gets smaller, and so that's going to make the thing get bigger on the whole. Of course, we have an s on the top as well, but that's multiplied by delta, by the inbreeding depression, that's less than 1. So overall, it makes k have to get bigger. Yeah? So self-fertilization in the co-sexuals, this S refers to the co-sexuals, makes it harder for a male to be able to invade. Now let's just look at the invasion of females, self-fertilization. What does that do to the small k that you need? And it makes it easier, that's right. And I showed a figure, and for some reason I seem to, in my lecture, I was going to show the same figure again to show those two, but I probably took it out because we were going to do it here. So self-fertilization of the co-sexuals gives females an advantage because they're avoiding inbreeding depression. That's what that equation is saying, that a little bit of increase in female fertility can be good if you also avoid inbreeding depression, and you wouldn't have inbreeding depression without some selfing. But in the male case, it goes the other way. Self-fertilization makes it harder for males. Why is that? What's the difference? What opportunities for mating are available if you increase your pollen output? Yes, the males get their contribution to reproductive fitness through the ones that are not self-fertilized. If you reduce that, you're reducing opportunities. Sometimes this is, the way people talk about this is, it's the genetic value of male gametes is reduced in a self-fertilizing population. They can only contribute to fitness through part of the female gametes, that part that didn't get self-fertilized before they got there. So although this is very, very simple, a as a model, it gives you an interesting insight. And I mentioned that in the lecture, that males are dependent on female gametes for their fertility and for their reproductive fitness. And so it's a more complicated kind of fitness than we're used to thinking about. It depends on the frequency of the other phenotype or the availability of their female gametes. And there's this asymmetry between males and females. It's a different asymmetry from the one with maternal transmission. This one is showing an asymmetry in whether males can invade, and is much more difficult, usually, except in the case of zero self-fertilization. It's much more difficult than females. Okay, so that was my first question. So what does it predict in biological terms? Yeah, it's easier. So we expect to see populations with co-sexes and females. We don't expect to see populations with co-sexes and males. That's what I said in the lecture. And that's true in the real world. Okay? And also I mentioned in the lecture that you can sometimes see those polymorphisms with co-sexes and males. And I mentioned that that's probably mostly because you had a case of dioecious population with pure males and pure females that was subjected to an environmental condition that selects for the females to get some male functioning. And then they would probably have rather poor male functioning. And so the males, the females could go back to being co-sexual, but they'd be pretty bad in their male functioning. And so males would have a much higher male functioning, and you could get this situation. Even though you couldn't get it as a mutation in an initially co-sexual population, you could get it when evolution is reversing and going back, because females will probably be bad male function. And that's precisely what we do see in the real world. Okay, that was problem one. Now we have problem two. I'm not sure whether we really want to go through too much of this because we want to get to Mike Whitlock's problems. I was now suggesting that we should do something a bit more complicated, apply the phenotypic fitness method to get the equilibrium frequency. Now, not of males. I don't want to look too much at males because these polymorphisms with co-sexuals and males are very rare, but polymorphisms with co-sexuals and male steriles, or females, those do occur, as I said, and I showed you a picture of a plant. So this time we want to assume that A1 is dominant, so we get phenotype A1, co-sexual phenotype, with either of those two genotypes, and females are due to a recessive male sterility mutation, like I said in the lecture this morning, and we had females being phenotype 2, just to make it absolutely sort of definite. And let now the frequency of females be X, and let's assume, like I said, that they produce 1 minus little k. So the questions are, we want to work out the equilibrium frequency, making those assumptions. And that's the first thing, is just to work out the equilibrium frequency. So it will be exactly the same sort of procedure we went through here, work out the female and male contributions to the next generation, taking into account the frequency when we calculate the male. We have to take into account the frequencies of co-sexuals and females, because males can get contributions to the next generation by fertilizing the female gametes of co-sexuals, whatever frequency they are, and fertilizing the female gametes of females at whatever frequency they are. so the first thing is to get the expression for the fitness, bits and pieces of the fitness, and get the two fitnesses. As I say, I think we want to do this quite quickly, so I rub these ones out so nobody is confused, because this was for males. It's a bit silly because for the co-sexuals, nothing's very different. Rub this out. So for the female contribution to the next generation through female fertility of the co-sexes, what do we have? Yep. So that was the outcross through female and the self-fertilized. Yep. And for the female, what do we have? Sorry, yes, sounds right. No S. Females they have no male gametes, so no self-fertilization, right? Yep. Yep. They just have that. What are we assuming here? That this is their seed output. We're assuming that all their ovules get fertilized, right? We're assuming pollen is not limiting. That would make a difference, of course, right? Let's just look at this one, the phenotype 2, the females. What is their contribution through male gametes? That's a nice easy one. And the contribution of the co-sexes through male gametes. They have S. Yep. 1 minus delta again, because they have inbreeding depression. And then they contribute through outcrossing. Through outcrossing on phenotype 1. Right? And its frequency comes in here, right? Plus outcrossing on phenotype 2, on females. What do we get there? No, the females, female gametes are always outcrossed, so it's not 1 minus S, it's just 1. I don't know if you said that 1 plus, because that's the amount of them. And then we have to divide by 1 minus X, I think. Oh, we have to have an X, nearly forgot that. The frequency, 1 minus X. So I'll leave you to do the algebra, I think, because we're running out of time. We want to get to Mike's problems. But we can come up with an equilibrium frequency from making sure that we end up with the same reproductive fitness of both phenotypes. Right? There's just a few more sort of thought questions. Brian, I think, mentioned in an earlier lecture that a recessive mutation is very unlikely to, an advantageous recessive mutation is very unlikely to invade a population. Why was that? Yeah, homozygotes are very rare, I think I heard. Why is it different? Why are we happy to assume it in this case? Yeah, because of the co-sexes are inbreeding. So if a co-sex individual in a population has a mutation that makes males sterile, because of the inbreeding, you would get some homozygote. Selection would see them. So that's, or you shouldn't think this is an inconsistency with what Brian said. It's perfectly okay in this population where there's some inbreeding. What else has been assumed? Well, this could probably go on a while because we assumed all sorts of things. I already mentioned we assume that females get fertilized. What other things have we assumed that have been mentioned earlier as things people assume in simple population genetics models? Not random mating exactly, but we've assumed the pollen pool is producing a sort of random mating, the contributions in the pollen pool. That's true, but it's not overall random mating, of course, because the co-sexes are partially self-fertilizing. There's a mixture of random mating and outcrossing for them. We've assumed non-overlapping generation. We've worked out the fitness as the gamete contributions in the progeny from the parent generation. We haven't worried about the possibility that this could be an organism with overlapping generations, of course. And I think I'll skip the last one. Why can males sometimes invade a population that comes to this equilibrium that's polymorphic? Because I mentioned that in the lecture, and this was just really making your minds go over the same thinking again. But I hope you'll remember from the lecture what's different about a polymorphic population when you're thinking about invasion by males compared with the one we just looked at earlier. Okay, I think we should go on to the other set of problems and not spend all day on these ones, unless anybody has any questions now before I hand over to Mike. But you see how simple it is to do these things with this simple sort of approach. There's nothing there except elementary algebra and making sure you don't forget some of the terms that should go in there when you write it down in the first place. Okay, all yours, Mike. Shut this computer down.
That two randomly chosen alleles in that population descend from the same allele copy in the immediate previous generation. 1 over 2n. That's right. So there's you choose one at random. The chance that the second one came from the same allele. There's 2n of those possible alleles, so 1 over 2n chance that it's the same one. Right. So that's good. Okay. So let's part A easy. We have 40 minutes left. Part B. So now let's use F, capital F, sub t to be the probability that two randomly chosen alleles in generation T are identical in state. Okay? In other words, if you looked at the DNA sequence of those two alleles, they're identical. Okay? They're not necessarily identical by descent. They are identical in state. Okay? So when you look at them, they look identical. They may not have had a recent common ancestor. Okay? ACCCT for one and ACCCT for the other one. They look the same. Right? We don't know how they got that way, but we don't care. They are the same. Okay? So identical in state, have the same DNA sequence. Okay. So using the same assumptions as before, that we have an ideal population, no selection, no migration, no mutation. What's F at time t plus 1, the next generation? Okay, so let's take a few minutes and just everybody work on this with your neighbor or something like that. We'll come back and talk to each other for a second. So we're trying to write the basis of a recurrence equation. What's this value f in the next generation as a function of what's the value in the previous generation? they are both copies of one of the parent alleles. Well, let's say this one has sequence ACCT. Well, we haven't allowed mutation. There's no mutation. So this one has sequence ACCT. This one also has to have sequence ACCT. Okay? So they're identical in state. Once we allow mutation, which we will in a minute, they may have been different. But without any way to change them, they're just perfect copies of the same thing, so they are equal to each other. Okay, is that good? So we have a probability that they came from the same parent times, if they came from the same parent allele, what's the probability they're identical in state? That's 1. Okay. But that's not the only condition we might have. They may have also come from different parent alleles. So what's the probability they came from different parent alleles in the previous? Right. So 1 minus 1 over 2n. Now, if they came conditional on they came from 2 different alleles in the previous generation, what's the probability that they're identical in state? FT. That's right. So that's what FT means, is that if you randomly choose 2 alleles, different alleles, what's the chance that they happen to be the same? That's the definition of FT. Okay, so that's our recurrence equation. That's the whole thing for this. That's the answer to this part. So it'll go up from one generation to the next. Okay. Let me just show you something really, get rid of all this mating system stuff. So a useful thing about this one, if you subtract both sides from 1, you get 1 minus f at t plus 1. You get 1 minus 1 over 2n minus 1 minus 1 over 2n ft. Okay, I multiplied both sides times minus 1 and added 1 to both sides. So 1 minus both sides. You get this. Well, that's also equal. Here's this 1 minus 1 over 2n that we can factor out. So we got 1 minus 1 over 2n times 1 minus f t. Okay, so that gives us this nice equation where we could just ramp this up to the power of however many generations we wanted to go, right? So if we wanted to go t plus 7 generations, we could just raise this 1 minus 1 over 2n to the 7th power to get the answer, right? Okay. So this 1 minus 1 over f, that's the probability that things are different. That's like the heterozygosity. Okay. Okay, let's go to part C. Okay, now we're gonna add mutation. Okay. Every allele, and we're going to add a very special, easy mathematical kind of mutation that is a good approximation in some cases and not so much others. But we're going to assume that every new allele that appears is unique. It's never been seen before. It's not present in the population. Okay? It may have been seen before, but it's not there now. Right? This is what we call the infinite alleles model. Every new mutation is unique, and so in that case, we don't have to worry about new mutations, being like any other part of the population. Okay? So this becomes a silly approximation if you look at a single base pair. It becomes a reasonable approximation to reality if you look over a sequence, because there's so many different kinds of mutations that could happen in that sequence that the odds of getting exactly the same thing are low. Okay, so but that's our approximation that'll make our lives much easier here. It's called the infinite alleles model, because in principle there's an infinite number of alleles. Everyone's new that appears. Okay. So now I want you to write, and I'll just tell you this and then again break off and let you guys do this. I want you to write a similar kind of equation as this, except allowing for this mutation. Okay. So let's did I say it up here? Let's say that the mutation rate is mu, just so we can all use the same variables. Okay. Is it clear what the question is? Just write a recurrence equation for this. But include the possibility of infinite alleles mutation. Go ahead. two alleles, and we're asking, are they the same? So what we've had so far is saying, what's the probability that they're the same after the drift, and accounting for the fact that they may have already been the same. But by adding mutation, we've got two new individuals. In order for them to stay the same, this is the probability that they're the same, not counting for mutation. In order for them to still be the same after mutation, the first one has to not mutate, and the second one has to not mutate. So probably the first one doesn't mutate is 1 minus mu. The probability the second one doesn't mutate is 1 minus mu. So to get the answer, we can just take this whole thing. Just take this whole thing and multiply it times 1 minus mu squared. Okay. And that's the whole deal. Right. Okay. Any questions? I talked to most of it one-on-one about it. Any more questions about how this goes? Okay. So now, here's our recurrence equation. And I want to just take it two last steps, 2 steps in one here. And I want to find what's the equilibrium value of f. And I'd like you to take it to an approximation that assumes that mu squared is small. Right? We're talking about mutation rates being very small, so square it. We really don't care about mu squared. We want to get rid of that. And that mu over 2n is small. Okay? So 2n is large enough that mu over 2n is a small thing times a small thing. Okay? So make those kind of approximations and try to find the equilibrium value of f. And what we'll see is they look very similar. They behave very similarly, and we'll get a 1 over 4nm plus 1 tomorrow when we talk about that. So we won't have to derive that. It's a very similar thought process. You make any questions? Great. Well, thanks, you guys. That was a lot of fun.